Hello dear students. In today's lecture, we will be studying the most amazing and famous experiments in the field of biology, that is, laws of inheritance. These experiments were so exemplary that they totally changed the direction of biology. When these experiments were performed by Gregor John Mendel in the years between 1856 to 1863, and he published his work in 1865, he was not given any importance because his work was way ahead of his time. Later on in 1900, when his work was rediscovered again, then people realized how exemplary, how wonderful was his work. And because of this path-breaking experiment, he was honored as the father of genetics. So today we will be studying all about his experiments and his laws. Okay, so he performed the experiment, as you know, on pea plant, Pisum sativum. And this pea plant itself was an amazing plant because he performed his famous artificial hybridization experiment on Pisum sativum because this plant was showing so many contrasting traits, contrasting characters, like he chose 7 characters. You remember it, isn't it? These 7 characters are stem height, flower color, position of the flower, color of the seed, seed shape, then shape of the pod and the color of the pod. These were 7 traits or the characters he studied. And he saw that all these characters had contrasting traits. Like for example, in case of the height, plant were either tall or dwarf. Color of the flower, flowers were purple or white. Then seed shape, they were again round and wrinkled and so on. We have already studied in previous lecture. Today we will be studying what experiment actually he did and how did he analyze these experiments. I will be using certain terms when I'll explain this experiment. Let's find out the experiments he did. He actually performed 2 types of artificial hybridization experiments. First experiments were called monohybrid cross. In monohybrid cross, he observed only the inheritance of one character at a time over various generations. He bothered only about one character and he ignored the other characters. Okay, so that was called monohybrid cross. Then he took any 2 random characters and observed the inheritance of those characters over generation, ignoring all other traits. Fine. So in monohybrid cross, inheritance of only one character over the generations. In dihybrid cross, inheritance of only 2 characters over the generations. And based on these, he got a lot of data. And this data was analyzed. And based on the analysis of the data, he formulated the laws. And these were 3 main laws. First was law of segregation and second was law of dominance. These 2 laws he proposed on the basis of monohybrid cross. And on the basis of dihybrid cross, he proposed the 3rd law, which is called as law of independent assortment. So today first I will explain you the monohybrid cross and the 2 laws. Followed by that I will explain the dihybrid cross and the law, that is law of independent assortment. So let's begin with the first experiment, that is monohybrid cross. Let us now study the monohybrid cross. As you already know, Mendel was also a mathematician. He formulated laws based on his mathematical calculations also. So, he assumed every character as one variable and he used the term unit factor. Today, these unit factors are considered as genes. But at that time, the concept of gene was not known and he used the term unit factor. So when we talk about a tall plant, capital T is a unit factor to denote the tall character. Likewise, for denoting dwarf character, he used the variable small t. This was assumed by Mendel. And because this is based on the sexual reproduction where two parents are there and the cells are diploid, so every factor is present as a paired allele. The term allele means alternative form. Okay, so when he talked about the tall plant, initially he took homozygous plants. Homozygous means pure line, which means the two alleles are of same type. So when we say homozygous tall plant, this means it is expressed by 2 capital T. Okay, and this one dwarf plant, he expressed it with 2 small t's. So here the parents he took were homozygous, means having pure character, no blending of characters. So initially when a tall plant which is homozygous and denoted by capital T is crossed with a dwarf plant having the variable small t, they will undergo the meiosis, that is reductional division, and they will produce the gametes. These are the gametes. These gametes are always single, which means they are segregating from each other. That is law of segregation. So from the parent, when gametes are produced, the paired form is segregated and every gamete has a single allele. So when you talk about this tall plant, only one type of alleles are present. They are possible. So all the gametes which are produced, they will be of only one type, that is capital T. Likewise here, when this small t undergoes meiosis, that is reductional division, all the gametes which will be produced will be only of one type, that is small t. And these gametes, when they fuse, fertilization takes place. So all the plants which are produced in F1 generation, F1 stands for filial generation. Filial generation means all the offspring of one particular generation. So they all were tall. But these plants which were tall, they were different from these tall in terms of their variables. Here it was denoted by capital T, whereas here they were denoted by capital T, small t. They are called as homozygous alleles, whereas these are called as heterozygous alleles, that is hybrids. Okay, so in F1 generation, all the plants were tall hybrid. Now these F1, the offsprings which are produced in F1, they are undergone self-pollination. And self-pollination means both male and female will be of same type, that is capital T, small t, capital T, small t. So in this case, two types of gametes are possible. That is, one type of gametes having capital T and other type of gametes having small t. Same here also, two types of gametes are possible, capital T, small t. So you can see that the offspring which are produced, this is a crisscross method, that is tree method. But this method is more easy, which is called as Punnett square method. On one side, we write one type of gamete. Suppose these are male gametes, that is the male gametes. And this side we are writing the female gametes. So when you see the cross, this type of combinations are possible. That is capital T, capital T, capital T, small t, capital T, small t, and small t, small t. And when he expressed all this data into the ratio, into the statistical form, he got these two types of ratios. First ratio was phenotypic ratio. In phenotypic ratio, he got that out of all, 3 parts were all tall and 1 part were dwarf. So he got the phenotypic ratio as 3:1. 3 types tall and 1 type dwarf. But when he analyzed the genotypic ratio, that is the type of variables which are present, then he got the ratio as 1:2:1, which means 1 part were homozygous tall, 2 parts were heterozygous tall, and 1 part were dwarf. So this is called as the genotypic ratio. So monohybrid phenotypic ratio is 3:1 for all characters, be it the height or color of flower, color of seed, shape of the seed, anything. It will be the same. Okay. Likewise, when you talk about the genotypic ratio, it will be 1:2:1. Okay. 1 part homozygous, dominant or particular expressing, and 2 parts were heterozygous, dominant, and 1 part were homozygous, recessive. Okay. So this was one trait, one character, that is stem height. The same thing we can do with another character. Let's see. For example, if we see the flower color. Suppose we denote the flower with purple flower and white flowers. Purple flower will be denoted by capital P, and white flowers will be denoted by small p. If they are crossed, so you know they will be producing only one type of gamete, that is capital P. They will be producing only one type of gamete, that is small p. And when they fertilize, they will be producing capital P, small p. That is, all the flowers will be purple. They were purple in F1 generation, first filial generation. And when you self them, selfing means self-pollination. So we will take capital P, small p here. Now they will be producing 2 types of gametes. One will be capital P, other will be small p. Likewise here also capital P and small p. And when you do Punnett square, so you know it will be, suppose this is male, this is female. These are signs for male and female. This is for male gametes and this is for female. So here it will be capital P, small p. Capital P, small p. This will be capital P, capital P, capital P, small p, capital P, small p, small p, small p. So this will be purple, this will be purple, this will be purple, and this will be white. So phenotypic ratio will be 3:1. Likewise, when you talk about the homozygous purple, this will be 1. Heterozygous purple, this will be 2. And white will be 1. So this is the phenotypic ratio. And this will be the genotypic ratio. Phenotype means whatever is observed, that is morphological observation, is called phenotype, and the genetic makeup is called genotype. So phenotypic ratio is 3:1 for all the characters, all the 7 characters where Mendel studied, and genotypic ratio, the ratio was 1:2:1 for all the 7 characters. So based on all these first monohybrid cross, he proposed 2 laws. First law was law of segregation. According to law of segregation, it states that during the gamete formation, the paired alleles, they segregate from each other, and the gametes have only one of the paired alleles. Gamete is never having paired alleles. Okay, so it is also called as purity of gametes. Law of segregation is also called as purity of gametes. Here purity means no hybrid character. Okay, so first law was law of segregation. So law of segregation states that during the gamete formation, the paired alleles separate from each other. This was first. Then second, which is the most famous law of inheritance, that is called as law of dominance. According to law of dominance, the character which expresses in F1 generation is called as a dominant character. For example, when we crossed tall plant with dwarf plant, in F1, all the plants were tall. So if I ask you, which is a dominant character out of tall and dwarf? It is the tall character. So any character which expresses in F1 generation or which expresses in hybrid state is called as a dominant character. And the character which is not able to express itself in hybrid state, that is F1 generation, but reappears again in F2 generation, that is called as recessive trait. Okay, so according to Mendel, through monohybrid cross, the second law he proposed was law of dominance. So according to law of dominance, the dominant trait expresses itself in F1 generation, and the recessive trait is not able to express itself in the F1 generation, but reappears itself in F2 generation in the ratio 3:1. Okay, so these are the 2 laws based on monohybrid experiments. Now we will study the dihybrid experiment, where Mendel took 2 characters at a time. On the basis of dihybrid cross, where Mendel took 2 characters together, he proposed the third law, that is law of independent assortment. We will come to this law later on. Let us first understand what is the dihybrid cross and what type of ratio it gives. So in case of dihybrid cross, any 2 random characters out of the 7 characters he took. For example, if we take a plant having round seeds, which is a dominant trait, but green, which are green in color, which is a recessive trait. So first type of plant will have the genotype capital R, small y. Okay. And other plant showing wrinkled seeds, but they are yellow in color. Wrinkled is a recessive trait, whereas yellow color is a dominant trait. So the genotype will be small r, capital Y. So any 2 random characters can be taken together, whether it is dominant or recessive, but they should be homozygous. Both the alleles should be same. Here both capital R, both small y, and likewise both small r, both capital Y. Okay, so these were the parents. Then in F1 generation, this is F1. So in F1 generation, he got all the hybrids because they will be producing through meiosis the gametes, and the gamete can have only one combination. Here capital R, small y. Here small r, capital Y. And when they fuse, they will be producing only one type of setup, that is capital R, capital Y, small y. And all these plants will be having round yellow seeds. Okay, so they were all F1 generation plants. Now comes the selfing of F1. So selfing of F1 means both the similar genotype are crossed. Capital R, capital Y, small one plant. Likewise here also capital R, capital Y, small y. And they are crossed. But here in this case, how many possibilities are produced? Here there are 4 possibilities are there. The gametes may be capital R, capital Y, one type of combination. Capital R, small y, small r, capital Y, and small r, small y. Likewise here also capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Okay, so these 4 combinations are present both in case of male and female. So based on these 4 combinations, this checkerboard, that is Punnett square method. This is called checkerboard or Punnett square method. It is very easy. On one side we will write all the possible gametes of male, and on other side all the possible gametes of female. So here we can write the same thing here. Capital R, capital Y, capital R, small r, capital Y, small r, small y. Likewise here also the same thing is written. Now see what combinations are possible. When they are combined, we can write capital R, capital Y, capital Y. So they will be round yellow, this combination. When capital R, capital Y, small y, the combination will be round yellow. Here also capital R, capital Y, round yellow. Capital R, capital Y, small y, round yellow. Okay, again in this row if you see capital R, capital Y, small y, again round yellow. But here in this case, if you see here, it is capital R, small y. Here the combination will change. Round, but this small y stands for green. So this will be round green. Again, here capital R, capital Y, small y, round yellow. And here capital R, small y, here also round green. This will be again a change. Now if you see here, capital R, capital Y, round yellow. Here also capital R, capital Y, small y, round yellow. But here small r, capital Y. This will be again a different combination. This will be wrinkled yellow. Same way here, small r, capital Y, small y. This will be wrinkled yellow. Here again, capital R, capital Y, small y, round yellow. Here, the capital R, small y, round green. So, it will be this combination. And here, small r, capital Y, small y. So, this will be this combination. So, there are 4 types of combinations are possible. One are round yellow. Second combination is round green. Third combination is wrinkled yellow. And fourth combination is wrinkled green. But their ratio will be round yellow. They are 9. 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 16. Likewise here, round green are 3. 1, 2, 3. They are 3. Likewise, wrinkled yellow, they will be 1, 2, 3. here. And wrinkled green, only one is possible. Okay. So this is called as dihybrid ratio. We don't go into genotype because it becomes very large. Okay. So for your syllabus, for your level, it is only the dihybrid phenotypic ratio, which is 9:3:3:3:1 for any two random characters. And based on this ratio and this dihybrid cross, Mendel proposed the third law of inheritance, which is called as law of independent assortment. According to this law, if any two random characters are taken together, they pass on independent of each other in the next generation. They assort independent of each other. They express independent of each other. It doesn't mean that one character will be only with one type of trait. For example, here round green seeds. Round and green are two different traits. So the seeds may be round or green, the seeds may be round and yellow. Same way, wrinkled and yellow, round and yellow. So any two traits, if you take together and see their inheritance, they express themselves independent of each other in the next generation with the F2 ratio. This is F2 ratio. Second generation, second familial generation. In F2, they show the ratio of 9:3:3:1. So these were two types of crosses, monohybrid and dihybrid crosses, and we studied three laws of inheritance, that is law of segregation, law of dominance, and law of independent assortment. I hope you have understood these laws very well. Thank you very much.